It's a real pleasure to be here. The green one. Okay. Great. We got that sorted. Angela talked about the central dogma. What I want to talk about is the ribosome, obviously, this magical molecule that can turn inanimate instructions in DNA and RNA, the nucleic acids, and turn them into protein. And within a couple of years of Crick coming up with the central dogma, the field had pretty much settled upon the idea that the ribosome is homogeneous and universal. One ribosome to rule them all, it can translate any messenger RNA. But little bits of evidence over the years suggest there may be slightly more to the story than this. If you look in various different ribosomal proteins, some of them you can knock out and they have quite specific effects. The Drosophila minutes are one up here. There's also some human diseases, ribosomopathies, where you have mutations in specific ribosomal proteins, and this causes diseases like black band anemia. Much less appreciated is ribosome variation that occurs in the RNA parts of the ribosome. Ribosome is made up of two components, ribosomal RNA and ribosome protein. What I'm going to talk about today is variation occurring within the ribosomal RNA, and in particular using zebrafish as a model. Before I get on to that, I just want to quickly stress why this is at the frontier of what we're doing. In genomics, there's been over 1 million people sequenced apparently, so you would wonder, what's left to do. But the truth is that 8% of the human genome was only sequenced as recently as 2022, and this includes all of the 4 ribosome sites that you find in humans, right? They're in different loci. We don't know if it's important that some are in different loci and if these different loci do different things, okay? And part of the reason why we know so little about these ribosomal DNA clusters, i.e. the regions of the genome that encode for the ribosomal RNA, is they're extremely hard to manipulate. There hasn't been a single knockout of a vertebrate ribosomal DNA that I know of, and consequently we have very poor model systems. All right? So this is why I want to draw your attention to the humble zebrafish. It has this really interesting dual translation system whereby we have a regular canonical ribosome that you find active in the soma, and I'm going to refer to the region encoding this as 45SS or 45S for the different RNAs that come off that ribosome transcriptional unit, okay, and S for somatic. And what we find is that as we go through development of those major different RNAs as they get expressed, going from egg through to adult, what we see is there's this transition. We have gradual increased expression of the somatic ribosomal RNAs, and they're encoded on chromosome 5, but we also have this 45S maternal or 45SM I'll be talking about, encoded on chromosome 4, and it's hugely expressed in the egg and gradually gets lost as we go through development through to an adult. The other thing to say is that these, unlike the different ribosome clusters that we have in human, these are really quite different from a sequence perspective, something like 86% in common. So still quite some differences. So what do they do? Is the big question. One really interesting aspect of the 45S maternal is that it's hugely amplified in the female germline. And when I say amplified, this is actually the DNA that gets amplified. It pops out from repeats flanking the 45S region, and it circularizes and it amplifies itself up like a plasmid via rolling circle replication. So that if you do sequencing in the female germline, we were doing bisulfite sequencing, but any type of sequencing, what you find is something like 1 in 5 molecules of DNA that you get back will be coming from this locus. And it's not just because it's a repetitive array, so quite a large locus, much more than just what I've represented here, but actually it's amplified extrachromosomally. So, what does it do? We decided we would have a go at knocking it out. I set Dr. Donna Bond and now Dr. Tim Moser to go ahead and try and knock this out. We used a relatively simple CRISPR approach where we targeted the promoter with 3 different guide RNAs, and we wanted to see what would happen if we knocked out this ribosomal DNA locus. And this is the results. We'll just focus on this up here for a start off. Because we're targeting a repetitive array, it was quite challenging to think of a system so that we could kind of get a metric on how many mutations we were making. So what we did was we set up a consensus sequence and we compared, like we did an amplicon sequencing over the regions that we targeted, and we compared it to a consensus. So when we look in uninjected fish, we run this amplicon test, we find that it's almost 100%. Remember, it's an array, so there'll be some nucleotide differences, but it matches the consensus as given by this reference conformity score 95% of the time, something like that. But when we targeted it, we found that we could go as low as 25% reference conformity. So almost, or the majority of cells had disrupted, somehow mutated 45SN. So this was great. We could show that we were doing something to the DNA. Did it matter at all? Did it affect these fish? And what we found was that the fish grew fine, really did. It was amazing. There was no difference in length. They swam around in the tank. You couldn't tell the difference between the ones that we'd targeted and the ones that we hadn't. But the amazing thing was, compared to the controls where we would get roughly 50% females often, or around that, when we targeted, we had 0% females. So by knocking out this locus, we basically halted female development. And they instead shifted into males. We've since gone on to have a couple of more generations of these fish. This is the original F0 generation that we targeted. And you can see in this individual we had a few different classes of mutations. Remember, we targeted in 3 different spots so we can create a series of different mutations. These were just used as a way to sort of track what was going on. This was the original targeting that Tim Moser did a couple of years ago now. We've since gone through some generations, handed over to a new student. In the middle, we weren't able to collect quite enough data of the F1, but in the F2, now that Connor has taken up this work, what we can see is that those mutations are being passed on and they appear to be relatively stable, separating into these different classes. So we've basically got alleles now that are segregating, and we can start crossing and characterising these mutants a little bit further. Here's another one. These had a single class, and again they're moving through that population in a fairly stable way. All right, summary so far. ribosomal DNA can be edited in a vertebrate. We've managed to do it with these fish at the specific 45S maternal or feminine rDNA loci. Those fish almost always develop as males. You can't make a female. There's no other detectable phenotype. The males, mutant males, produce sperm just fine. They can mate with wild type to give rise to healthy F1 females, and males and females. And like I say, those mutations appear to be stable. One thing I just want to quickly touch upon at the moment is how important is that sex phenotype, the fact that this loci appears to help contribute to be essential, at least for feminization. And this is interesting because in zebrafish, despite being intensely studied by a medical model, we don't actually know what's the trigger of sex determination. Particularly in wild strains of fish, so don't worry too much about these different strains here, but the top two strains are domesticated strains, with the bottom four being wild strains. And if you've got really good eyesight, what you'll notice is that on chromosome 4, right at the end of the chromosome, we have a high linkage to between sex and this locus, which is on the end of chromosome 4. So what we think is that this could actually be the mysterious unknown gene which determines sex within zebrafish, but we haven't done the sort of right overexpression experiments, complementation experiments to prove that just yet. So what are some of the other emerging questions that come out of this? What does this RNA locus actually look like, rDNA locus actually look like? Despite having worked with it for quite some time now, we still don't actually know how many repeats there are, in the same way that it took until 2022 for us to figure this out in humans. We wanted to study this using long-read nanopore sequencing. We first started by looking in clones that we could get from the original genome assembly and did long-read sequencing on that. And what we found was that on one of these backs there was this really clear signal, TTA GGG, which is of course the telomere repeat sequence. So we realised that this region is actually right at the end, literally right at the end of chromosome 4. And what that meant is that we could then use a telomere probe to pull out the very end of all the chromosomes and then sequence back in this direction to try and assemble as much of these repeats as possible. And indeed, this is what we've done here. We can show something like 6 to 18 repeats. It appears that there's some kind of strain dependency, and we're looking to see if, for example, repeat number correlates with the likelihood of developing into females or males. So this is all work that's underway. Hopefully I can talk more about it soon when we've confirmed a few things. What are some of the other questions that emerged from working on these interesting loci? We're very interested in other species. In particular, in New Zealand, we've got this spotted wrasse. It's a fascinating fish that lives in a harem on the rocks around New Zealand. If you take away the male in this harem, then the largest female will turn into a male. And as it does, and it reprograms its gonad, we wanted to ask, is there a difference between the males and females with regard to the feminine ribosomal DNA? So we looked at that. We managed to find that there's this huge trace of amplification of one particular ribosomal DNA. So it appears to pop out just like the zebrafish, do the rolling circle amplification and create lots and lots of copies in females specifically. And we can quantify this as shown here by the percentage of reads mapping to the ribosomal DNA locus that we're interested in, 45 SM. And just like in zebrafish, we were seeing up to 1 in 4, 1 in 5 reads coming from this ribosome locus, so massively over-amplified in females, whereas the males, which obviously there's much less of them, they don't do this amplification. They have their gonad turns into a testes as they come into the breeding season, and therefore stay as males. All right, a quick summary of what I've told you. There's a hell of a lot to learn about ribosomes, variant ribosomes, particularly in the fish world, but it may also be true that there's interesting things to find in humans and mice, and I can talk more about that with anyone who's interested. What I've told you is that in zebrafish, we can knock out this alternative sex-linked ribosomal DNA and block female development, but affect no other part of fish development. We've found that it's also possible to assemble these loci, and we can do this in a relatively high throughput way using this telo-seq approach, where we pull down from the telomere to look at this particular locus. And when we sort of port this out to other species, we're starting with spotty because it's an interesting wrasse from New Zealand that we can get our hands on. It seems like there's similar principles despite having a unique system. They have different repeats, different sequences, but it clearly amplifies and does some of the same things. Other species are exciting too that we're looking at, like salmon and carp, etc. And with that, I'd just like to acknowledge the people that did the work, in particular Donna Bond, Tim Moser. Connor Tumulty has just taken over from Tim, and Oscar did a lot of that early work to get the model going. Erica, Neil and Simon are helping out with the spotty work. We have a zebrafish facility in Otago that's very good. We've been funded by the Marsden Fund, and also I just wanted to again say, like everybody else has been, thank you to HFSP for their courage continuing to fund fundamental biological science, and also for their ability to give courage to others. I think when I was doing my fellowship back in 2009, I needed a fair bit of courage to retrain, but the fact that I was forced to by going on to this fellowship has given me a lot of fortitude to go into different areas, which I'm doing with this ribosome work. Thank you.